presentation. Today I'm going to talk about exploring neuroimaging signatures where hypothetical representation learning. Actually, there is some overlap with the previous speaker. How about now? Here.
Can you project your, like, we now see the both slides, that's the problem. How do you project that? I'm not sure. I never used that. So you see what's going on, right? So you have both, all of that. So can you project?
Yeah, but so
online it is good. So,
yeah,
okay, so let's do it. It's okay.
Okay, so yeah, so there is some actually overlap with the previous speaker, Ricky, Tianqi Chen. So, so today my talk will including four parts. The first, I will give a little bit of the introduction and background, and second, I will introduce the different graph embedding techniques, including two different Euclidean space. Embedding method is basically embed the graph into vectors and also embed a high-dimensional graph into density Gaussian distributions. And also the third section, I will give a presentation on hyperbolic graph embedding for human brain network analysis, mainly for two different applications: subjective cognitive decline prediction and also the normal brain aging trajectory detection. And lastly, I will give some conclusion. So first, because I want to give a little bit some background about the human brain disease, like Alzheimer's disease has all of these. So AD is a progressive brain network disease with very complex etiology and multiple pathogenesis. So by 2050, there will be 152 million dementia patients and also 1.1 trillion healthcare costs in the world. And if you look at this figure, actually, sorry, this is so small here. If you look at this figure, you can see that actually before the onset of AD, we can see actually before the detection of the AD, actually there is almost 2 decades earlier already the people already developed AD, Alzheimer's disease. So starting from 40 years old, actually when they're 70 years old, actually they was detected as Alzheimer's disease patients. So there are 3 major biomarkers for Alzheimer's disease. So 2 different proteins, alpha beta and also tau protein. And the other one is a very important one is the brain connectivity alterations. Basically, we can use the very advanced neuroimaging modality. This is a machine we call magnetoencephalography. We call MEG neuroimaging modality. So we can use this to measure the brain activity directly from different patients. So and another topic application we are working on is for the normal brain aging trajectory detection. As we know, our brain is a very core part of our human body, and it also is a seat of the intelligence and the interpreter of the senses, and also an initiator of the body movement and also controller of behaviors. And our brain are affected at both the structure and functional levels by normal healthy aging. And also, we can see that the National Institute on Aging, they announced actually will continue to support the research to identify age-related neural changes and mechanisms that older people use to maintain optimal learning, memory, and the cognitive functions. But currently, the studies about the trajectories of functional level brain connectivity alterations in the healthy brain aging are still not fully understood. It's very limited. And in our project, we aim to study normal brain aging trajectory by examining alterations in the hierarchical structure of the brain networks, including both the brain regional level and also subnetwork level changes over around 600 subjects between 18 years old and 89 years old. So with actually different imaging modalities like T1 structure, MRI, and also MEG functional neural imaging data. So this is a brief introduction about the neural imaging. Actually, modalities fall into two broad categories: structural neural imaging and functional neural imaging. So we basically now work on focusing on the functional neural imaging data, MEG data. Basically, the MEG and the functional MRI and also EEG are all functional imaging modalities. So they can measure the brain activity temperatures, and also for the like diffusion MRI also is a functional neural imaging. They can measure the molecular diffusion in the biological tissues. However, the structural neural imaging will describe the shape of the size and the integrity of the gray and white matter structures in our human brain. So based on both the structure and the functional neural imaging, we can actually build the brain network. Basically, we call brain connectome based on the collected data. So we can, but we need to use some brain atlas. We also call it brain templates, which actually has some predefined brain contour regions. So we can actually, based on the brain atlas, to extract the brain regional level temperatures, and then we can compute the brain connectivity matrix based on the adjacent matrix for the graph. And here we can, because our main focus is on the MEG neural imaging data, actually we can compare with other functional neural imaging modalities. We can see that it has a very good temporal resolutions. Actually very excellent is in milliseconds compared to the functional MRI. And it was invented by a professor at MIT in 1970s. And another very important property of MEG neural imaging is they can measure the absolute neural activity directly. Compared to functional MRI, actually they can only measure the relative neural activity where the oxygen of blood flow near active neurons. So this is indirectly measured. And also MEG, when they collect the data, they can be recorded in sleeping subjects, and it contains less noise compared to functional MRI. That's some the background. Now we are talking about what's human brain network. So human brain network, basically based on the collected neural imaging data, we can build the human brain network with different brain regions and different nodes in the graph. And also we have the connections between different regions. So in the human brain networks, actually they include several modules and also hierarchical structural nature. So it's different from the unstructured, the 2D images and also the language data. So each graph node has a different number of neighbors and also has arbitrary and varied graph structure and sizes with different number of unordered nodes. So another important is non-Euclidean space and with complex interactions between the different nodes. And so how to effectively and efficiently learn high informative representation for the very complex human brain networks and for early detection of the Alzheimer's disease. And so next I will introduce some different graph embedding techniques. So we can use these techniques for human brain network embedding. So the first one actually is the existing some graph representation learnings starting from the 2016. The node to back method actually is motivated from the word to back, is proposed from Stanford group. And they can project the graph node representation as a low dimensional vectors in the Euclidean space. And the other type of approach is graph to Gauss, is published in 2018, and also there are some similar works that they can encode the high dimensional graph into density function space-based Gaussian distributions. So they can actually quantify the uncertainty in the latent space for each graph node. And in recent years, I started from 2017, actually the graph neural network, graph convolution neural network becomes more and more popular and the attention networks. However, This also learned from the representation or edge representation as vector in the Euclidean space. And now, due to the large language models becomes very popular, the graph transformers model also, there are quite a lot of works on that. They can address the two main issues of graph neural networks, the overswitching and the long-term dependency, but with the conjective computational cost due to the self-attention mechanism. However, there are actually very limited methods can capture the node level hierarchical property in the level space. But this is very important for different downstream tasks, especially in healthcare and finance and transportation and many other domains. So this is a little bit summary of my previous work on stochastic graph embedding for static graph and temporal graphs. So we have done some static functional brain network embedding into Euclidean space before, basically to encode the brain network into a lower dimensional Gaussian distributions to quantify the uncertainty information, basically how many subnetworks in the human brain networks, using the variance of the Gaussian embeddings. So these are our two previous papers having in 2020 and 2021. And on the right panel, it shows the dynamic graph embedding with recent works, 2022 and 2024. This year, January, we published around 2 papers on dynamic graph embedding. And so this slide will introduce the static graph embedding into a Euclidean space for the early stage Alzheimer's disease detection. Basically, this is not in hyperbolic space, but in Euclidean space. But so this is for the real data set. We have 76 mild cognitive impairments patients. There are actually 48 stable MCI patients and also 28 progressive patients. Basically, they will convert to dementia after 2 years. And we also have some normal control subjects, 53, for the classification tasks. And the imaging data is MEG data is 5 minutes and 306 channel. So basically, based on the collected 5-minute sensitive MEG data, we use the brain atlas, including 68 brain regions. This is the regional textures data, and we can compute the information to generate the adjacent metrics about the brain connectivity. And then we develop an approach based on Gaussian embeddings. We can embed this brain connectivity into lower dimensional Gaussian distribution in terms of limit and variance. And based on this embedding, we can actually do different downstream tasks. The first one, we actually do the classification for AD progression prediction. And second one, we actually want to detect, as shown in this slide, we want to detect which brain regions with significant FGG network alterations between the different subgroups. For example, the very important two subgroups is the stable MCI and progressive MCI. Why those people convert to dementia after 2 years? So this is the significant brain regions with significant alterations detected based on the embeddings we learned from the neural networks, and it's largely included in the temporal and the frontal regions in the cortex. And constantly with the previous study using other different neural imaging data, especially the temporal lobe, we have the hippocampus, which is very important for the Alzheimer's disease. And we can actually store our memory. So basically, it means in the temporal lobe, actually, they have a big alterations between the two different subgroups. And on the right side, it shows the classification results based on the different groups for the classification. We do the normal controls and the stable MCI and progressive MCI. And here, in the next following slides, I will start to talk about why we want to choose hyperbolic space and the embedding space, because a lot of complex and real-world data that actually exhibit underlying hierarchical structural property, for example, brain connectome and genetic state of protein network and ontology networks and genetic network and so on. And Euclidean space has very limited space for embedding like train, like graph data has very high running time and memories. However, the hypothetical space as shown here is negative curvature, this one. Compared to the Euclidean space with a zero curvature, you can see that actually they can actually preserve both graph distances and complex hierarchy extremely efficiently in very few dimensions. So on the right side, it shows the Poincare disk actually Ricky Chen just already presented the Poincare disk and also hyperbolic remaining manifold. So they can actually are very can effectively embed those scale-free graphs. So into and with a very high like exponential growth space actually from the radius. We can see that this is the current node of the binary tree. And the XY of the children nodes, we can see that actually the red curve shows the hyperbolic distance and the pink curve shows the geodesic distances and the blue curve shows the Euclidean distances. So we can see that hyperbolic distance can actually approximate the geodesic distance in the hyperbolic space. And also, so here, this slide, we can show that more clear. So this is the Euclidean space to embed the tree-like graph, and also this is the hyperbolic space to embed the tree-like graph into a hyperbolic space. We can see that in the Euclidean space, actually the nodes will overlap and also clustered together. So it requires higher dimensions in the embedding space to embed more nodes. However, in the hyperbolic space, it grows exponentially with the hyperbolic radius and much close to the boundary of the Poincare disk. We can see there are more space to embed those children nodes. And also it has a much lower distortion actually, as all the nodes can be separable in the latent space and require very small embedding size. So the important thing for us to study very network is the hierarchical property. So this is some of the fundamental mathematical formulas for the hyperbolic space representation models. There are two very typical models for the hyperbolic manifold representation. Is Poincare model actually proposed in Meta company in 2017 and 2018. Dr. Nico. I think, yeah. So we can see that compared to Poincare disk, they have a very different the distance formulas here. This is the dp xy, this is the dl in the Lorenzo model. So the distance is much simpler, so it's numerically more stable and simpler formulas, and more efficient remaining optimization, and it's scalable to larger datasets. And here the key is the curvature, negative curvatures here. And here in the hyperbolic space, we cannot use the stochastic randomness and optimization. Immediately, we need to use another optimizer. It's called the remaining SGD optimizer. Basically, we need to actually map the features from the based on the exponential map from the tangent space with manifold and also to manifold from manifold to tangent space use a log map. So this is how to compute the gradient of the parameter CT, and the eta is the learning rate. And here is the remaining SGD optimization, how to update these parameters. And this is the objective functions to in the remaining manifold space. And here shows the hyperbolic embedding compared to the Gaussian embedding I used to work on for the brain networks. So hyperbolic embedding, we can see that this is from Dr. Nichols' paper in 2017 for the WordNet database dataset. So we can see that in the central of the Poincare disk, we can see the word is more generic and has a high hierarchy, and also goes through towards the boundary. We can see to the location and to the city and to New York. It's more and more specific and has a lower hierarchy. So this is a very important property for us to study brain networks. That's why we want to choose this approach to study the brain disease and also the aging trajectory detection over time. So next, I will present two recent projects. First one is using the hyperbolic graph convolution neural network. Actually, the first initial paper is published in 2019 from the Stanford group. So here we work on the MEG data using the AAL brain atlas, including 90 brain regions, to learn the brain nodal embeddings for different brain cortical regions. And here, this is the output, and we can see that actually the triangle basically indicates the left hemisphere regions, and the circle represents the regions in the right hemisphere. So we can see after it is split to different sides in the primary disk. So this is symmetric. Actually, this is a good validation for brain network because we have a left hemisphere and a right hemisphere. So this is very initial because not many papers using hyperbolic embedding for brain connectivity. So in the very beginning, we need to validate this approach works for brain connectome. And so in our project, we have 49 subject to cognitive decline patients. So basically, those patients will have 5 times likely converted to Alzheimer's disease. And we also have another conversion group, is 41 clinical normal subjects. So this paper is now under revision. So it's already on the preprint, it's on the bioRxiv. Yeah, we got very good comments recently from the actually very good journal. And here is a comparison for the experimental results for the link prediction task with 3 different baseline. Basically, it's a standard graph convolution neural network in the Euclidean space and also temporary embedding. And another is hyperbolic graph HJC model with the fixed curvature. And also we also study the curvature as the learned parameters. We can see that based on the learned curvature settings, the C, capital C here is a learned negative curvature. You can see that actually with a fixed curvature, actually the learned curvature can get actually better results, better performance. And also in the another findings based on this result is also consistent with the very initial paper from 2017. Is that hyperbolic embeddings, we can only need fewer embedding dimensions compared to the Euclidean spaces JSON. And we can see that they need embedding dimension 8 to achieve 82% accuracy. And here we can only use actually dimension 3 to obtain 95% accuracy for link prediction tasks. So finally, we choose the 3 dimensions with learned curvature C to generate the brain regional embeddings for the downstream task that we want to study. What's the difference between the hierarchical changes in different subnetworks? So here, this is the dorsal attention network, and also this is the ventral attention network, and this is the sensor motor network. So the blue bars show the healthy control, and also the red one shows the subject in cognitive decline. We can see that actually the SCD subjects has a very decreased hierarchical radius, hyperbolic radius in the latent space. Basically means when the radius decrease, basically it means they lose more connections, and the brain connections, basically the node degree will reduce. And so for the healthy normal control people, we have a more stronger connectivity because maybe due to some dead neurons and so on. And also this is some relations with hierarchical alteration to cognition based on some other iterations. So the 3 subnetwork with pronounced alterations between the 2 subgroups actually correlated to some higher scores on the depression scale. And the central motor network also has greater impairment in completing daily function activities, and also dorsal attention has a higher subjective rating of memory failures. So these are very important detection for study the SCD prediction. And this is we study because you will want to ask, actually the brain network is not the largest scale graph, why you just compute based on the original adjacent matrix is a small matrix. Based on that features, basically this is the blue, this is a histogram, and the blue histogram, the bars show based on the original adjacent matrix being the classification result. And however, the green and the red bars show based on the embedded features and also combine the embedded feature with the original adjacent matrix features. We can see actually the hypothetical features capture partially unique features and improve the classification scores over the original adjacent features. And the second project we want to talk about is about the very recent work, actually very recognized experts, is on the human brain aging trajectory on 600 healthy patients across the 7 decades, age decades. So from 18 years old to 89 years old. So we have the MEG neuroimaging data, and also we want to detect the changes in regional level and also subjective level across time. So this is our data set from the Comcom data set. So we have 7 different decades from very like a teenager to like more older people, 80 to 90 years old. And we have and this is how many female subjects. Basically 50% female and 50% male, approximately. So in total, we have 587 subjects. And the imaging data is still the functional MEG neuroimaging data computed based on the very atlas HCP is a very public one, including 360 brain cortical regions in brain. And so this is a physiological value basically is a metric for the brain connectivity estimation. So this data set is from our collaborator in Italy, University of Trento, actually. They have a MEG center there. And so in this data set, we also have other multimodal features like cortical thickness, basically based on the structure MRI data set. They can mirror the thickness of the cortical. And also we have another myelination. This is also very, as demonstrated, is a very indicator for aging, human brain aging. So we have 2 different modality features, and also the MEG neuroimaging brain networks. And we input these node features and also the brain adjacent matrix into the hydraulic graph convolution neural network to generate the human brain network embeddings. And this is we want to quantify the hydraulic radius changes over age decades. 7 different age decades, and every age decade we have around I think 100 subjects, less than 180 subjects, around 85. So this is for the right early visual cortex. We can see that actually the hypothetical radius increasing over aging decades. So this is basically reduced hierarchy of the right early visual cortex during aging among this 587 subjects. And this is more detailed results based on the RI, basically the regional of interest level hierarchical changes across age decades for 6 different subnetworks. We can see actually the interesting finding in this project is some sensory and motor subnetwork. We can see actually they maintain the hierarchical radius actually maintain until 60 years old, and after 60, 70 years old, it exponentially grows. Basically, the connection loses after 60 or 70 years old. However, like this one is a primary, so basically, is a very important area in our brain. And it's like a linear increase, basically the hierarchical decrease linearly. So it's like a heterogeneous changes over time. And this is a cortex hypothetical radius for different subnetwork level. And the different color parts show the different 7 different age decades. So the purple shows the teenager 18 to 30 years old. And then we can see actually some basically we compute the slope for this each subnetwork. And this is a final result based on subnetwork level hierarchical gradient slope. So basically the subnetwork is a larger slope value have more pronounced hierarchical changes beyond aging. We can see the primary so early visual cortex has a higher hierarchical changes over aging. However, the early auditory and posterior ocular has a relative like unchanged hierarchy. So this is more studies about in the hypothetical space, basically the embedding space. We study the different, we compare the first decade, basically the 18 years old to 30 years old. And for the early visual, basically we find that have increased average hypothetical radius, basically decrease the hierarchy, lose more connections over aging. And this is the 7 decades when the people are between 80 years old and 90 years old. We can see this is the hypothetical radius increase after the 7th decade. So when you get a like aging over aging, so the early visual cortex basically decrease the hierarchy, lose more connections due to some diagnosis. And this is the early auditory. We can see in the hypothetical space they didn't change that much. Basically remain unchanged between the first age decade and the 7th age decade. And this is a visualization in the 3D human brain for the left hemisphere, this side, and right hemisphere. So this is the early visual subnetwork region. Basically, is in the occipital lobe. They can process some images and is a very important area. And here, the sensory and motor is a second subnetwork we found has a increasing lobe here. A larger slope is a decrease the hierarchy. So these are the very important regions. Basically, the subnetwork level hierarchical changes for the aging human brain aging trajectories. And lastly, I want to thank you all my great collaborators at MIT. This way, I work with 2 master students for their master thesis in 2021, 2022, and 2023. They are graduating now and working in the Amazon robotics and some startup. And also thank you my collaborator from Madrid to provide the data for the SCD, the first project, and the second project, the data provided from the University of Trento in Italy. And also thank you other collaborator from the Johns Hopkins and the Houston, the University of Texas Health Houston Center. And my DOE grant. And this is all the references about this graph embedding work for the brain networks and 2 for the dynamic graph embedding networks. Okay, thank you very much. Thanks.
Two questions. Next all. Also, there is some fresh copies and if you want to browse. Okay.
Yes.